Yeah. Yeah. Thanks for the introduction. It's a tremendous privilege for me to be here today. I want to talk to you a little bit about the kind of work we do in the laboratory, and I also want to tell you a little bit about my story. And also, my emphasis will be on, careers in science. I'll use mine as an example. And I just want to reflect on the idea of adopting or pursuing a career doing research in science. Okay, now that could be in medicine, could be in physical sciences, could be in social sciences, whatever your passion is. One of the themes of the story I'm going to tell you is that I've enjoyed a lot of good fortune. But I think what perhaps you can see that that it's important to as your career proceeds is to really avail yourself of the people around you and really learn from the people around you. And as you'll see, I benefited enormously from the people around me. Okay. So I'm not going to talk so much about Argonaut or the microRNA. I'll talk a little bit about microRNAs, but my emphasis will be a little bit more on the journey, And basically, my objective here today is to encourage you young folks who are thinking about science to go with it, I have a Polish connection, and that's why I'm here today, because my Polish friends have invited me, And the connection is my dad was born in Poland. It was in a little village in what was Eastern Poland, which is now in Belarus. And he and his sister were orphaned at a fairly young age, and they lived with relatives in the region around their village or in their village. But Longin ended up going to high school, and in those days, in the 30s. In Poland, not everybody went to high school. In fact, my dad said that in his village, kids went to the 3rd grade, but 6th grades was mandated so they would repeat the 3rd grade, right? So you really had to have a certain amount of opportunity to even go to real 6th grade, right? And he was able to do that because he was identified as talented by his teacher. And his teacher, saw little Longin. I guess he was, diligent and he read a lot or something. I don't know what she identified, but she identified him as talented and enrolled him in a national scholarship program to support the continuing education through the 6th grade and through middle school, and then finally high school for talented youth. And if it weren't for that scholarship program and that teacher who enrolled my dad, and if my dad didn't pass that exam to get into gymnasium, I'm sure I wouldn't be here today. There he is in Wilno with some of his buddies, but he only went 2 years because the war started in 1939 and the school closed. And then he went back to his hometown and home region, but he had to live kind of on the run because there was a warrant for his arrest, that the Soviets had, because his uncle, with whom he lived, was the town clerk, and the uncle actually was sent to Siberia, and Longin, avoided being captured, but he was captured by the Germans. And then in 1942, he was sent to a work camp, basically, a hospital, factory in Dudenhofen, Germany. And he spent 3 years there. And actually, because he had gone to gymnasium and he had studied German there, and then I guess in those 2 years, he became fluent in German. So he became had a leadership role amongst the other Polish people in his camp as a go-between, And then he was liberated by the U.S. Army and came to, I forgot something. In our audience is Georgia Kordovich, right, who is the son of Victor Kordovich, who founded that scholarship program. He established that program. And so I owe a lot to your dad, Georgia. Yes, my dad moved to Vermont and married an American woman, and there I am. These two people, my mom and my dad, were interesting people. My mom was a writer, and my dad, as you'll see, he was a woodworker. But their business initially was farming, dairy farming. So they milked cows, and I learned to milk a cow. So I grew up on a farm in Vermont, and I think one of the most important aspects of my childhood is I was allowed to just wander around. I was actually in a large family, and they didn't pay much attention to the kids, and so I remember my childhood as basically I had some chores, but mostly I just wandered around and just explored, right, and amused myself. So I ended up doing a lot of projects, a lot of them, supervised by my dad. But I remember basically becoming interested in science so early that I can't remember not being interested in science. Actually, I'm remembering something in this moment that was important to me is that I remember my dad was, only went to high school for 2 years, but he was very well read. So we thought that he was very knowledgeable, And so I remember I would ask him questions all the time. I know you pester your parents, and parents get pestered with questions. And, I remember that, his answers were always really inspiring. But in retrospect, I realized I don't know if he necessarily answered my questions correctly, but it was the fact that he was there and gave me time, it was what I remember very warmly. So I think that my journey in science was really supported by my dad and my mother, even though we were just living out in the country. So the kinds of things that I wondered about, I remember very much thinking about things like, why are all maple leaves the same shape? So I used to really wonder about things that are very common and right in front of us, and that must have been what I would ask my dad about, and like why all birch leaves are the same shape, and why are they different shapes, and how are they different shapes? How's a leaf, what? So we know this is genetic, right? Because a birch tree makes a birch tree, and a maple tree makes a maple tree. So in that seed that makes those trees is the information to make a tree, and they're always this same type. It's unique. So those instructions are in the DNA. And at each little bud where you make a leaf, there's information to make a leaf of that shape, right? And it always does it like that. So this was the kind of I think prompting that that led me to think about developmental biology, right? And when I went to MIT to college, a lot of you are getting ready to go to college. The main thing that I remember that was so important about my experience in college was, number one, the amazing things I learned from my fellow students, right? And which was a surprise to me. I thought we would learn everything from our professors, right? And the other was the opportunity to do research in laboratories was there, and you had all sorts of opportunities. And so this was a very valuable experience because here I had a mentor, in this case Ed Gruberg, who really cared about my training and taught me sort of the method of science, right? How do you explore a question? How do you find a question, formulate a question, design an experiment? And then we actually wrote a paper and published a paper together, as an undergraduate. So I learned how to do that process. So I would encourage young people to really look for those opportunities to do hands-on research and, and do it. It's an amazing sort of, confidence building experience, right? Then I did a PhD with David Baltimore. And then postdoctoral research is what a scientist does after they get their PhD, because usually you're still thinking about what you want to explore, what kinds of biological questions, in my case, biological questions I want to study, let's say, going forward in my career. And the postdoctoral research gives you an opportunity to work with a mentor, in this case, Bob Horvitz, to learn the kind of skills you want to use in your independent research as when you become a professor and start teaching and leading research groups. And that's where I learned to work with this nematode, Caenorhabditis elegans, which I'll tell you a little bit more about. And so then I had a faculty position at Harvard, Dartmouth, and finally UMass Med, where I am today. And during that whole time, I've worked with this C. elegans nematode. I can't talk about my career without highlighting the person who makes it all possible, which is my wife. And we married in 1976, so we're going to be our 50th year this year. And Rosalind, or Candy, has been my lab manager and collaborator continuously since 1985 or so. And most importantly, she and her coworker, Rhonda Feinbaum, actually conducted the research that led to wherein they discovered the first microRNA. So that's the research that won this Nobel Prize, which I shared with Gary Ruvkin. So as you can imagine, I have an infinite, a debt to Candy. So defining influences in my lab are, Longing, playing on the farm, MIT, and of course Candy as a constant, and then these mentors, all of which, from whom I learned a lot, and then the worms that we've studied, and then the microRNA that we discovered. So, so my lab has been sort of captivated by this question related to the leaf shape, but how do animals get their shape? And, if you just think about it, it's pretty amazing. Chickens are like chickens, ducks make ducks, and then rabbits and insects are like crazy bizarre shapes of all these animals, including insects. But what unifies, and then C. elegans, which is a lovely little worm that lives in the dirt. I'll show you some examples of it. The organism's DNA contains this information that will cause the elaboration of a very specific developmental program, whereby the egg and then the embryo turn into the adult. So the unique final shape and functionality of that animal is the result of a unique developmental program, and that's somehow encoded in the DNA. And so that's the sort of the fundamental mystery and amazing suite of phenomenon that we want to study. How is it that the information for making an animal, like this animal, right, you all, how is it contained in that tiny egg? And then how is it elaborated? And how does it do it without making errors, right? So we can study this problem using animals that are convenient for experimental purposes. And the reason why this is powerful is because we have a lot in common, us animals. Even though there's an amazing diversity, let's say, amongst all these animals here, these are just examples of bilaterally symmetrical animals. What unifies them is they have actually a genomes that we sequence the genomes so we can count the genes. And there's about 20,000 genes in all these organisms. Actually, whether it's the nematode with 900 cells or a human with trillions of cells, we have about the same number of genes, right? And we share actually a significant portion of those genes. In other words, as you compare genomes across all these animals, you can spot about 10,000 genes that are really essentially the same. They're making the same proteins. Okay? So if we think about our common ancestor of all these bilaterians, this was about 500 or 600 million years ago, okay? And, transport yourself in your mind back 500 million years. There was an animal there. There was an animal then that gave rise to all of us, right? And so we're really amazed by how all these animals can have so much in common, but be so distinct in form and function, right? And they are using, for the most part, almost the same set of genetic machinery. Those genes were present in the bilaterian ancestor. And as evolution flowed, all this diversity could arise, all from that same ancestor. So we really are interested in two things at the same time. We know that the genes, shared genes unify us. So if we study shared genes in one organism, we can learn about those same genes as they apply in the other organisms, right? Also, we can, by studying the genes of different organisms, we can try to learn more about how they are made to be different. How does evolution work, right? So here's the human, and then the model organisms that have been traditionally used to facilitate studying these shared molecular and genetic apparatus in cells are, the fish. You've heard about them, melanogaster, C. elegans, and also there's a flatworm, and another fascinating acial worm, which I'm going to talk about and mention at the end. So this model organism idea was one that was originated basically back in the 1970s, initially with Drosophila work, and then C. elegans, which also adopted starting in the 70s. And the idea is that if we study genes of this model, we'll learn, that will facilitate understanding human biology. And the reason why is we share a lot of genes. So here's, here's an opportunity to play a little movie. So you get the sense of what it looks like, to look through the microscope, just a dissecting microscope. You guys have dissecting microscopes here in your labs. I don't know if you've worked with, has anybody worked with C. elegans in this school? Well, I'm here to, your life has now changed because now you're going to, it's a wonderful animal to work with. It's about 1 millimeter long, the big ones. It's crawling around on a petri dish. You just spread some bacteria on there, and they eat the bacteria, and they grow. You can see those oval things. Those are the embryos inside an egg that's been laid by this adult. The adult is a hermaphrodite. It makes both sperm and oocytes internally, so it fertilizes internally, so it's very convenient. But there are also males, so you can do crosses and make new genetic combinations for experiments. And you can see the larvae crawling around, and what's quite amazing about this animal is the life cycle from embryo that you see there to an adult that's making more embryos is about a little bit over 2 days. So you can set up an experiment in your lab on Monday and get the results, on Thursday. Get the progeny on Thursday. So we just love this animal. It's just endlessly fascinating and contains and reveals, if we know how to explore it, it reveals a lot of fascinating biology, and biology that applies broadly. And the microRNA discovery is an example. So get ready for this, okay? If you've never seen an embryo developing in a video, speed it up a little bit. You're ready. I think you're ready for thrill. And because, especially if we think about what's going on and think about how marvelous that is, right? Okay, so this C. elegans embryo is poised to develop. It's a little deceptive. It looks like it's 2 cells, but it's actually, it's a 1 cell, and it still has the sperm and the oocyte embryo, nuclei not fused yet, but they will quickly fuse in this video, and then you'll have 1 cell, and then it's going to go to 2, then it's going to go to 4, 8, and so forth. Okay, so, and all the cells, whoops, come on, man, don't do that. Okay, so there's 1, 2, 4, 8, 16. I just love this. See, they're getting smaller. They all have nuclei. Those are the flat things in the middle. They're kind of like fried eggs, right? And those are the nuclei. Every nucleus has the exact same DNA, right? But somehow there's instructions being deployed from those nuclei in different cells differently, so they rearrange themselves into a worm, right? And it has a head, a tail, it has a pharynx, a brain, intestine, muscles, skin, and it actually has only 550 cells, right? So that's a whole animal in just 550 cells. But of course, what we're fascinated by is, well, how does that happen? How does the DNA in one cell and the identical DNA in its progeny cells, up to 550 cells, how does that cause the production of all these different cell types and arranged properly in this animal? And this is really fundamentally the phenomenon that underlies all animal development. It just, C. elegans is exemplifying it for us and giving us the opportunity to study it. So let's think about what's going on, okay? On the left, we have the single cell, and on the right, we have that embryo at the end. It's just before it hatches out from its egg. 550 cells, but they all have the identical DNA, so how can those cells be different? You know, we can identify neurons, and actually what we find is neuron-specific genes are expressed, and genes that are not supposed to be on in neurons, like genes that are expressed in muscle and intestine, are not on. And in muscle cells, I'm pointing to a muscle nucleus, muscle genes are on, right? Those you guys have learned about myosin, actin, troponin, you know, genes whose proteins who are expressed only in muscle, and they function there, and the things that are on in neurons specifically aren't on. So there's this really amazing deploying of differential gene expression, and that's regulated somehow. All the information for regulating gene expression properly during development, so you make all these cells in the right place in the right time with the proper constituents, you know, happens because of instructions in the DNA, right? So we know then that genes, so let's think about this idea of gene expression and differential gene expression. So consider two genes. This is the DNA. And, you know, the DNA is transcribed into RNA, which is a copy of the sequence of nucleotides from the gene. That RNA contains the information that can be translated into a protein, right? And that protein, because of its sequence of amino acids, will fold up into a structure that has amazing, specific functional properties. For example, it's evolved to make part of the cell, or maybe it's to help make bone or something like that, right? So we can think of this as a structural protein, or it could be a protein that's evolved to do work, like an enzyme, right? And the other gene makes a protein, and this protein is evolved actually to regulate other proteins, right, or the production of that protein. So this we call a regulatory gene, and then this can be considered an effector gene. And in general, our genomes contain, a significant portion, which are making gene products that have evolved to regulate the production of other gene products, okay? And so that's basically how the animal manages to deploy gene expression properly throughout all the different cells of the animal. And it turns out that, to foreshadow that the microRNAs that we discovered turned out to be a new kind of molecule, not a protein, that regulates the expression of the genes. So we came upon the microRNAs by doing genetic analysis of C. elegans, and the logic behind that is that people wanted to discover genes that are regulating development, right? These would be, and therefore we'd like to identify the genes and then learn how they work. And the idea is that if a regulatory gene is mutated, then the regulation of development would be abnormal in some way, and so you would get an abnormal worm in some fashion. And so the mutation that changes, the development is going to lead us to ultimately to the RNA and presumably protein products of that gene. And so we can now connect mutation to gene product to a function of some kind that's orchestrating developmental processes, right? And so the mutants we studied were these basically developmental timing mutants, okay? And one is LIN4. So you can see that this animal, I think you can see, if you compare it to the normal, which is the top adult, this animal looks a little peculiar. I don't know what that means. Yeah. Okay, yeah. I thought that was like a gentle alarm to say, you should stop soon, or something like that, yeah. So the wild-type hermaphrodite is at the top. And wild-type means all its genes are intact and not mutated. And the one that differs from the one at the top, it only differs in one gene, and that's the Lin4 gene is actually deleted. And so these animals, because of the absence of that one gene, they're missing many parts of the body that are usually produced in the later larval stages. And that's because the animal repeats the first larval stage over and over, right? It's actually a fertile animal because the gonad develops normally. And remember, I told you they can self-fertilize. So this is a viable, if I could bring to you this mutant and you guys could propagate it in your lab, biology lab, indefinitely. And every generation you could look at it and say, yeah, it looks peculiar. And you'd notice that it never lays its eggs. And the embryos that hatch out always hatch out inside the mother. And then they consume the mother and then crawl away. So we call it a matricide phenotype. The Lin14 mutant exhibits an opposite defect. It skips early larval stage developmental programs. And so parts of the animal that would be made in the first larval stage, for example, are never made because the stem cells that produce those parts skip immediately to making later types of cells. And they actually the whole animal becomes an adult sooner than it should. So we call that a precocious mutant. And by just thinking about these mutants, without knowing what their gene products, you could come up with a logic diagram, which is that the animal does transition from juvenile to adult in many cells of its body. And Lin14 is very important for that transition because Lin14 promotes juvenile programs and inhibits later programs, and Lin4 regulates Lin14, right? So Lin4 turns off Lin14 to enable the animal to make the L1 and then go on to the L2, okay? So again, this is just a logic diagram because we didn't know what the gene products were. So these two labs, the Ruvkin lab and the Ambros lab, set about to find the genes corresponding to those mutations that caused those defects, right? And what this meant was, the teams in each of our lab were essentially sorting through hundreds of thousands of base pairs of DNA to find the mutant sequence, right? And that actually in the early 1990s was a very labor-intensive process, and it would take a couple of people 4 years to find the gene, starting with the mutant. Nowadays, we have genome sequences online, so this kind of search for relating mutations to genes and gene products is much more straightforward. And so the Ruvkin lab found that the Lin14 gene, where the mutation is, it makes a messenger RNA, and that messenger RNA, when there's a mutation there, makes a mutant protein, which doesn't do what it normally does. But normally, it's a regulatory protein, and sure enough, it regulates juvenile genes, right? And then in our lab, Rosalind and Rhonda, or Candy and Rhonda, found the mutation for Lin4, and sure enough makes an RNA transcript, but the RNA is not a coding RNA. It does not make a protein, okay? It makes a small RNA of about 22 nucleotides long. And that was the novel discovery that we made. That was unexpected. It was unprecedented. Nobody had ever noticed or found a gene making a tiny RNA before. And so, but fortunately, it was actually almost immediately apparent once Gary Ruvkin lab and my lab shared their information about their respective genes. It was clear how Lin4 regulates Lin14 because actually it base pairs to the Lin14 mRNA through complementary base pairing. The Watson-Crick pairing, G's to C's, A's to T's. It's very specific. Although in RNA, it's A's to U's. And so that allowed the gene product of Lin4 to repress or dampen the production of protein from the messenger RNA of Lin14. So that was the novel discovery. It hadn't been described before. And so it was called a microRNA because it was so much shorter than any other RNA people had described. It's only 22 nucleotides long. But in that Lin4 was the first microRNA, and the second microRNA was discovered by Gary Ruvkun a few years later, actually, 6 years later, also in C. elegans, right? But what was incredibly striking about LET7 was those 22 nucleotides are perfectly conserved in the genomes of all the bilaterian animals, right? So the common ancestor, right, of all of us had the LET7 microRNA. And everybody ever since has had that LET7 microRNA with that exact same sequence. So it was important 600 million years ago, and it's been important ever since. So this microRNA discovered in C. elegans, immediately revealed itself as universally important in ways yet to be discovered at the time. And there's many other microRNAs discovered. First of all, Lin4 was found to be also across all the bilaterians. It's just the sequence is not perfectly conserved. Actually about at least 24 additional families of microRNAs apparently are involved in the common ancestor of all the bilaterians. And they were probably part of the expanded toolkit of regulatory mechanisms that enabled the bilaterians to be so complex. We have far more complex internal organs, far more complex nervous system. We have appendages, which appear in all sorts of forms across these animals, right? And it's thought that the microRNAs were actually an innovation that appeared in the bilaterian ancestors, and it facilitated the kind of diversity that this group of animals exhibits. Big message for you all. Discovery of microRNAs was an accident. We didn't discover microRNAs because we were really smart, or that we had great vision, that we had a way of seeing something that it had to be there, even though nobody knew it was there, right? There was no theoretical grounds for proposing that there should be this small RNA mechanism of gene regulation. So we stumbled upon it. And the notion here is to remind ourselves is that again and again, scientists across the planet are making discoveries, even in this moment right here. Why? Because they're really curious about the observations in front of them, right? Now, this doesn't mean it's always people studying C. elegans. It could be people studying the cells, related to Argonaut syndrome, and discovering phenomena that was unexpected and revealing new biology, right? So this is a very, very important principle to really keep in mind. And this leads us to have insights, having discovered the microRNAs, about microRNAs, in human disease, et cetera, et cetera. So my final little story is going to be illustrating in a direction the lab is taking now, where we're working not on C. elegans, but another little worm. It's called the banded panther worm, and it's an acele. It lives in the ocean, and it has a remarkable ability to regenerate its whole body. So if you cut the animal in half, or even if you're even more creative and crazy, you can cut it into many, many pieces. You can dice up this animal with a razor blade, and you end up with fragments. Now, in this simple case, there's two fragments, a front half and a back half. But over the next few days, those two halves regenerate a full body of an acele. And now you have two aceles with all their proper body parts. The back end, which had no brain, now has a brain, right? And notice they're half the size of the original one. So they're actually doing all this without feeding. They're using stored energy, And you notice they reconfigure their relative scale. So somehow the cells not only make new parts that were missing, the whole thing gets reconfigured into the right shape. And so people are studying these phenomena because they present to us principles of development, regeneration, and wound healing that apply generally. Let's think about, here's a mental experiment that I think helps illustrate sort of how marvelous this process is, right? So if you think about this animal before you cut it, it's soothing, actually, right? It has an anterior posterior let's say, map. And then people study this actually to find out, we know a little bit about the molecules that are deployed in this way. There's some molecules that are abundant in the head and then they decrease as you go tailward. And some molecules behave oppositely. So, and these molecules, in some cases, they can show that they actually can kind of direct the cells to kind of know where they are and differentiate accordingly. So that we call this an AP axis of information. Okay, and now consider a cell. So like get yourself into the mind of a cell in that, and you're like one of hundreds of thousands of cells in this animal, and there you are. And the experimenter comes cruising in with his or her razor blade and cuts behind you, okay? And a few days later, that fragment that you find yourself in makes a new flatworm with all its proper parts. And so the AP axis was restored, even though initially this fragment only contained, more anterior information. So that's one of the processes that we want to study, is how do you reconfigure a new AP axis of information. We also want to know how is it that the cells know how to respond, right? So because that cell there is in front of the cut, and so therefore, it's going to contribute to the production of the back end of the animal, right? Okay? So first of all, the cell gets some information. It hears something and says, damn, my neighbors are completely missing behind me, And then there's a response, and then there's a program of rebuilding what's missing, right? Okay. Let's think of an alternative universe where you're the same cell, but the investigator with a razor blade cuts in front of you. Okay, it's the exact same cell. The back end makes a worm again with a front end, including a brain, amazingly, right? And so the AP axis was regenerated, and that cell, you're the same cell, but you're now making front material, right? So the same cell is programmed very differently depending on the local information that it's receiving after the wound, right? And so I think you can understand that this feels fundamental, doesn't it? Doesn't this kind of remind you of kinds of things that happen, when wounds heal and so forth? And so we're studying this in collaboration with Mansi Srivastava at Harvard, and the basic idea is, what can we learn about wound healing, and regeneration more broadly amongst animals in general? And we want to study microRNAs in this process, and we've begun that, and it looks really promising. But before I do that, I want to let's meditate on this. Okay, the ACEL, the guy at the top that we just talked about, can do whole body regeneration. And planaria, famously, some of you may have worked with planaria, you can do the same thing. You chop them in half, and a few days later, or a week or so, you've got two planaria that are perfect body plan, right? Head, tail, brain, mouth, and everything, right? So what we know, though, from we've studied already, is some of the same genes and molecules are operating in the context of whole body regeneration in the ACEL and the flatworm, okay? So that means our common ancestor, there's an amazing bilaterian common ancestor. I'm here to, I want to change your life. So from now on, when you wake up in the morning, you think, it's good to be alive, right? And I'm really thankful for that bilaterian ancestor, good job, right? Also, it's a source of inspiration for trying to understand fundamental processes of evolution and development. So there was a toolbox for whole body regeneration in that ancestor. And so the obvious question is, well, what about humans, right? And of course, famously, let's say humans do not regenerate their whole body. It's really, even if you cut off a finger, you don't regenerate. Although once, now I'm reminded of something I didn't plan, but it reminds me when our first child was born, we brought it, he was just old enough to bring into the lab, so we brought him into the lab to like tea time, and to show him to colleagues. And my colleague, who's a developmental biologist, he says, if you cut off that child's finger, right, and you don't put a bandage on it, it can regenerate, right? And of course, this happens in places where people, know enough not to wrap it up and clean it up, right? And so the idea is that actually, so a young child can regenerate their finger. Anyway, so I'm here to tell you that human beings are very good at tissue regeneration, and we should be proud of it. And I can tell you this is my last slide. From my own experience, this is Ambros's thumb a few years ago. I'm a woodworker, I have a router, and usually I'm careful, but this time I wasn't. So there's like a huge chunk of my finger missing, and I thought I would document the process of this thing healing, you know, and perfect, right? You know, the fingerprint is restored. It made thumbprint. It made thumb skin exactly for that location on my thumb. It didn't make pinky skin. It didn't make elbow skin, right? It didn't make too much skin. It didn't make too little skin, okay? So, you know, this is the kind of everyday phenomenon that has to do with our own biology that presents itself to us. And it's frankly mysterious, right? Scientists, we know a little bit about how this works. We know some of the molecules. We know some of the signals that are happening. We can put together imaginary models that are testable by further experiment. But we cannot tell you how that works. Especially we can't tell you how it works so perfectly, right? And remember, our organs are regenerating themselves all the time. The skin, you may know, regenerates itself how many times during a healthy lifetime? If you live 70 or 80 years, you could replace it, I don't know what the word is, 15 times or something. Your intestine replaces its lining every few weeks, right? The liver regenerates, you know, and wounds heal throughout our body. So we have this amazing capacity to do this in a very, very, very regulated fashion. You know, the intestine generally doesn't make too much intestine. The skin, when it regenerates, makes just the right amount. So these are the, you know, I'm captivated by these sort of everyday biological phenomena that we can potentially access in an experimental system, you know, like the ACL. Final thing, it was not my last slide. See, I fooled you. The last thing is reflection. Why do we do scientific research? And you know, its utility, right? Basic science research, and physics and chemistry and so forth leads to engineering, you know, innovations, and the things that we care about so much. You know, the GPS in these things wouldn't work unless we knew about Einstein's principles of relativity. Those equations go into the calculations that actually locate this. Einstein didn't discover those equations and those principles in order to make GPS work for us today, right? That's just one outcome of that. Has utility. So understanding nature leads to better living. And this, you know, whether this is life science, climate science, you name it, okay? But the other thing I wanted to emphasize today is a sense of wonder. And so I want to encourage all you guys to really recognize that one of the most strongest, you know, human instincts there is a sense of wonder, curiosity, right? Amazement, and then the desire to learn more about that, right? Hey, 3.5 billion years of life on Earth, right? There's an enormous amount to study about the diversity of living things and how they work and how they interact. You know, 600 million years of complex animals like ourselves. And the final thing I want to say is that where we stand today, right now in this minute, most of what there is to know is unknown, right? And that's a fact that I know, that I'm convinced of, and I could explain, you know, lots of reasons why. But it is something we need to remind ourselves of because, why? Because this is what I believe, is that there's a strong human instinct to believe that in this moment, in this place, we know everything we need to know, right? Because what would life be if we were in a constant state of uncertainty about our surroundings, right? You know, I have faith in the construction of the building, right? I know that, you know, just crazy things are not going to happen. This will stay here, it won't fall, whatever. All these things in our macroscopic world that we rely on, we get comfortable in, and we understand everything around us, right? But that's an illusion based on our macroscopic you know, experience. So when you look out into the universe, if you're an astrophysicist, you know, you look down into living systems, there's layer upon layer upon layer upon of mystery yet to be understood and explored. Thank you.